Hello, everybody. My name is Nilufar Monhassari, and in the following 30 to 40 minutes, I'm going to present you our comprehensive solution for your regulatory T-cell research. Let's have a look at our agenda for today. First of all, I would like to give you an introduction about our company, Miltenyi Biotec, then talking about Treg research as a basis for adaptive immune therapy. Afterward, I will go through our complete workflow for Treg research, which includes sample preparation, cell isolation, cell activation and culture, and also phenotypic and functional analysis of your Tregs. At the end, I would like to sum up with Treg translational portfolio and offer you all the material and resources that you can use to get more information about our products and support. Let's begin with a brief company introduction. Welcome to Miltenyi Biotec, empowering discovery and advancing therapy. Our mission is to advance scientific research and medicine by providing solutions to curative cell and gene therapy as well as biomedical research. Miltenyi Biotec was founded in 1998 by Stefan Miltenyi, and it's the largest owner-managed company in Germany. We have two main sites in Germany. Bergisch Gladbach is our headquarters, and in Teterow, we have the GMP manufacturing facility. We have subsidiaries in 28 countries, distribution to 73 additional countries, and we are over 3,000 employees worldwide. And we are going to serve the needs in life science research and to translate new scientific findings into clinical applications. At Miltenyi Biotec, we cover different topics and support our customers with translational portfolio and workflow. And now let's have a look at Treg research as a basis for adaptive immune therapy. Tregs are important subset of adaptive immune cells and control immune reactions for maintaining homeostasis in the body. All the T cells develop in thymus out of a common precursor of cells. And depending on the stimuli, they can develop to CD4 or CD8 T cells. Tregs generate upon their encounter with self or non-self antigen and mediate tolerance or suppress aberrant immune responses. And Tregs classification based on development origin, you can see here, I just activate the pointer. In the central system, thymus-derived Tregs, which called T Tregs or naive Tregs. They are the majorities and most stable population of suppressor Tregs. We have P Tregs here, preferably induced Tregs in vitro, and they may convert to T effector cells. And we have I Tregs, induced Tregs in vitro by application of IL-2, TGF-beta. You can have Tregs in cell culture. And characterization of the Tregs. Looking at this facts panel, in the sense that there is no unique surface marker for Tregs, they are characterized by the combination of surface markers. In general, Treg cells express CD4 and CD25 as shown in the facts. And the more precisely, the highly expressed CD25 cells can be gated. Moreover, Tregs can be further identified by the absence of marker like CD127, and the naive Tregs can be identified by the additional markers such as CD45RA, and due to the facts, especially naive Tregs can only be identified by the combination of several surface markers. And these cells can be isolated if you want to have the very high pure cells, only by fellow sorting. And also, it's good to keep in your mind that CD8 positive Tregs are presenting as a hot topic in several research and studies recently. And the main functions of Tregs: suppression of overwhelming immune response, which keeps the balance and homeostasis in the body. They suppress the immune response against self-antigen and non-self-antigen, which again protect against autoimmunity and mediate tolerance in the body. And both of the first two functions are really like favorite for the body. But the third one is suppression of anti-cancer immune response, which support the tumor growth is not the favorite function of our body, and during the cancer treatment, this function of Tregs should be tightly controlled. Tregs has crucial roles in different types of diseases and provide novel therapeutic strategies for treatment of different immune disorders such as inflammatory diseases, tumor immunity, transplant tolerance, and autoimmune disease. And according to the key rules, it can be studied from different aspects in the context of wide variety of diseases. So my question here is, from which point of view are you studying your Tregs? You are in the research part, you are in a preclinical studies, you are in a clinical. Whatever your answer is, the good news is that Miltenyi Biotec is covering all aspects of a Treg studies. And I'm pretty sure that during this webinar, you get the answer of most of your questions. And if not, please contact us. Here, I would like to use this great publication in the Cell Journal that was published some months ago, which discussed about Treg and CAR-Tregs as the effective and novel immune cell therapy methods regarding to autoimmune disease, graft versus host disease, GVHD, organ transplantation, and so on. And I highly recommend you to read this publication. I'm going to show you an example in the next slide. Yes, this picture here shows that the healthy situation, there is a balance, immune balance between regulatory T cells and T effector cells. However, in some circumstances, this balance is disturbed, like GVHD, autoimmune disease, or organ transplantation, rejection, inflammation, and so on. And the reason which underlies immune imbalance in each case are different. And adaptor immune therapy with Tregs leads to the reestablishment of the immune balance. And it's the reason that why Tregs are very attractive candidate for cellular immune therapy. Let's have a look at an example. Here is an example of Treg therapy as an immune cell therapy approach regarding to solid organ transplantation. One of the underlying reasons for the rejection is often an inflammation of the transplanted organ. After organ transplantation, the immune suppressive co-treatment can either successfully protect the organ, in this case, for example, or the organ get inflamed because of the endogenous immune cell recognition. Immune cells that they recognize the new organ as foreigner and attack it. And it happens 17% of the patient in the first 3 years post-transplantation. In this case, blood can be collected from the patient and Treg cells can be manufactured in a clinical scales from autologous blood products. And then Treg as a treatment injected to the patient. And Treg can endly transfer to suppress and the ongoing inflammation in the patient's body. And in this area, there are several ongoing studies, clinical studies for the application of Treg or CAR-Treg as a novel immune cell therapy. And the application of Tregs and CAR-Tregs is not just limited to organ transplantation, but also in graft engineering and stem cell transplantation, in GVHD situation, or for example, in autoimmune diseases like diabetes type 1, they are highly studied and used right now. Okay, let's go one step further and have a look at Miltenyi Biotec workflow for Treg research studies. Just to remind you that we have a translational workflow and we cover whatever you need for research and clinical studies. But today's webinar is just about our research portfolio and optimize your sample preparation. The secret of successful experiment lies in your starting material because the quality of your sample preparation influences directly all downstream application. So my advice to you is that start smart. Tregs can be isolated from different tissues, tumors, and blood. Regarding to mouse, the main source of Tregs are spleen and lymph node, as well as tumor and other tissues. In human, Treg can mainly isolated from PBMC, but at the same time, it can be isolated from different organ, tissue, and so on. And it's good to mention that Tregs are very sensitive and fragile cells. So be aware that you need to be very gentle and careful during your sample preparation. So I would like to offer you gentle max dissociator and dedicated tissue dissociation kits, which combining mechanical disruption and enzymatic digestion in the most effective gentle way to obtain a viable single cell suspension from even hard tissues for downstream application or analysis. Gentle Max Dissociator is a benchtop tissue dissociator. It's programmable and has optional temperature regulation as well. It is really gentle to the cells, which lead to the viable single cell suspension with preserved epitopes. It is reproducible by using predefined programs for standardized tissue processing. Fast functionality of this device results in processing up to 8 samples in parallel with GentleMax Octo Dissociator and offer you a safe process in a way that sample processing can be done in a closed and sterile system. Here is an example. We applied Lamina Propria Dissociation Kit from mouse with the application of GentleMax protocol to prepare our samples for the next step of separation and so on. After dissociation of the tissue, we did the FACS analysis and checked for the Tregs population. And as you can see here, you can see a good population of the cells after dissociation, CD3, CD4, FOXP3 positive cells. And here you can see the comparison of the number of the CD4, FOXP3 positive cells out of manual method and GentleMax protocol. It's really, the difference is really visible. And also in this plot, we want to show you that the quality of cells out of GentleMax protocol is comparable to the manual method, and the membrane is completely conserved. And you can see the consistently high number of viable FOXP3 Tregs out of the combination of Lamina Propria Dissociation Kit and GentleMax protocol. Okay, our sample preparation product is not just limited to GentleMax and our dissociation kits. We offer you more options. For example, here, we have the Max Tissue Storage Solution, which is convenient to use. You can store your tissues up to 48 hours, and it's efficient. You will get no cell activation or apoptosis afterwards, and it's really flexible. It is tested for a variety of tissues type. And also the other highlight is Max's smartest trainers and pre-separation filters that help you to have no clumps, no aggregation in the final yield, and also your uniform cell suspension. And in this way, you can have most effective labeling afterwards in the separation step. And dead cell removal kit is a straightforward removal of dead cells and debris from your cells, especially during the time that you want to culture your cells. It's really important. And also it enhances performance of any sensitive downstream application as well. And here is another application example in which the skin tissue was dissociated with GentleMax protocol and GentleMax situ. Afterwards, it was removed, the clumps were removed using the smartest trainer. The size was 70 micrometer, and at the end, cells were analyzed. You can see here the total number of cells out of manual protocol and gentle max protocol. It's comparable, whereas we have less variation comparable to manual process. And then here you can compare the CD4 FOXP3 positive cells as Tregs. You can see the cell number in from gentle max protocol is much more higher than manual protocol. And here you can see this comparison with FACS analysis that you can see more FOXP3 positive cells out of gentle max protocol rather than manual protocol, which is another proof of the gentle max functionality. Okay, let's choose the best strategy for Tregs isolation. For isolation of Tregs, we offer you a wide variety of products like Treg Isolation Kits and MicroBeads, MACSxpress Treg Isolation Kit, REAlease MicroBeads, and cell sorting using MACSQuant TytoCell sorter. And I'm going to talk about each of them during my talk. In addition, you can benefit from manual to automated cell separation system, different device. To getting more information about each of them, please visit our webpage. But before choosing the best strategy for isolating your Tregs, you need to answer two questions. What is your starting material? Is it blood, tissue, or tumor? And what is your plan for downstream application? When you know the answer of these two questions, you can choose the best strategy for your Treg separation. As many of you might know, Miltenyi Biotec offers a great technology for cell separation, which calls MACS technology. Why MACS cell separation technology is so special? To make the story short, we have minimal labeling with no effects on cell biology for full downstream compatibility. In more details, the column amplifies the magnetic field by around 10,000 volts. This allows for minimal labeling and smallest bead size for only about like 50 nanometer. In consequence, less epitopes are covered on the cell surface and less stress is applied on the cell. So the isolated T cells Tregs are fully fit for downstream application and analysis. And it's good to know that MACS MicroBeads were even approved by the FDA to be used in clinics. Well, since we are expert in all technologies, we can play with them and use them for maximum efficiency for given need. For example, talking about positive selection with microbeads and microbead kits is the fast way to isolate T cells and especially interesting for the customers that want to optimize their workflow time. Furthermore, microbeads are ideal to directly isolate T cells populations that are defined by one specific marker. This makes them the ideal tool to use when isolating rare cells from heterogeneous and hard to process tissue with highest purity. Furthermore, after positive cell separation with microbeads, the negative and unlabeled fraction can be used for a second round of isolation, for example, another T cell subset and so on. And of course, looking at the price per separation cells, microbeads are quite an economic solution, to be honest. Untouched isolation. What is untouched isolation? You can isolate your cells untouched with the isolation kits. Works via depletion of non-target cells. This is necessary when the target cells fraction is not defined by certain markers, but rather by the lack of these markers. So all cells expressing defined exclusion markers are depleted. Isolation kits are to be recommended also as the ideal pre-enrichment strategy before fellow analysis or before sorting. And yes, let's go to the next step, MACSxpress solution. is to isolate human Tregs directly and without density centrifugation from whole blood. It is fast, easy, and convenient, and especially interesting for the industry customers who wants to save time. Max technology offers different manual and automated system separation system, as I've mentioned before. Manual separation with QuadroMax 4, it is shown here for 4 samples, and AutoMax Pro for walkaway convenient for up to 5 samples. And the Multimax solves 24 separation plus. You can process up to 24 samples in parallel. And the last but not the least, Multimax X for flexible, fully automated, high throughput industrial applications with any source of materials. Okay, let's have a look at Treg separation with Max Isolation Kits. We offer you different Treg isolation kits for different species with various downstream applications. But it's first on you to let us know what is your need, which Treg subset you are going to separate, what is your downstream application or test. And as soon as we get your answer, we help you to choose the best kit for your studies. For example, here, if you want to have highly pure and high recovery and quality of the cells, and you want to use the cells for in vitro suppression assay, we recommend you to use CD4 CD25 Treg Isolation Kit. Whereas there are some customers who are looking for the highest purity of FOXP3 positive cells with purity up to 96%, for example. In this case, we offer CD4, CD25, CD positive, CD127 dim or negative isolation kits, and so on about the other isolation kit. So my recommendation to you is that take advantage of this flexibility and choose your proper kit according to your needs. Here you can see the basis of our Treg isolation kits that is available for human, mouse, and non-primate. And I've chosen the CD4 CD25 positive Treg isolation kit as an example. And as I told you, our Treg isolation kits have more or less the same basis. And it consists of 2 steps: depletion of unwanted cells, positive selection. Okay, in the depletion of unwanted cells here, non-CD4 positive cells are depleted. All the cells except the target cells are labeled with MACSQuant. By passing through the magnetic column, the untouched target cells get released. You can see here. In the positive selection, for CD25 positive cells, cells of interest that are here, CD25 as I said, are labeled with MACSQuant. Then the labeled cell captured in the column, and then after removing the column from the magnetic field, cells can be eluted. And at the end, we obtain positive cell fractions. Here you can see the example in human and primate. You can see the efficiency of Treg isolation in the right side in comparison to the non-isolated cells. You can see the cell population and scattering of the cells in FACS panel easily. And here is the application example of CD4 CD25 positive Treg isolation kits. These kits obey exactly from the basis rule as I've shown in the previous slide. First, depletion of unwanted cells, and then positive selection. And the final yield out of this kit is really amazing. We will reach high purity and recovery. In addition, this kit is optimal for the researcher who wants to use both fraction of the Tregs and 2 responder cells. I mean, Tregs are CD4, CD25 positive cells, and 2 responder cells are CD4, CD25 negative cells. And it is important to have negative fraction as 2 responder cells for some downstream application like in vitro suppression assay that I'm going to talk about it in the very near future in some minutes, and you will see the application and the positive point about these kits. Okay, I hear from lots of you, especially who are working with mouse tissue, that you have lots of different tissue at the same time, and automation is highly demanded. I would like to suggest you fully automated system, Automax Pro Separator here, which is the best option for you. It's fast, easy, convenient, and flexible regarding to analysis of several different samples at the same time. I would suggest you to check our webpage to get more information about this device. And we are coming to MACSxpress Treg Isolation Kit. Are you isolating your Tregs from blood and you need to be fast and efficient? If yes, here is your solution. MACSxpress Treg Isolation Kit for Human is the special kit which helps to isolate your Tregs directly from blood. It is one of the most easy, fast, and efficient methods, which gives you this opportunity to save your time by skipping the density centrifugation step. As you can see here, you labeled your cells and deplete the non-CD4 positive cells and then label the CD25 cells here, and then elute them and have your final yield. The whole process takes 30 minutes, as you can see here, and much shorter than the conventional isolation methods. And you will have excellent yield, you can see here, out of this kit. Our next Treg cell separation highlights are the REAlease MicroBead kits. This revolutionary microbead technology allows to positive select cells, but then remove the microbead and the antibody labeling to get completely label-free cells. REAlease MicroBeads kits are available for many human and mouse T cells and non-T cells, including CD3, CD4, CD8, CD19, CD25, and so on. And the process itself is quite easy. After cell separation, there is a bead removal step with the bead release reagent. Cells are now ready for a second round of, here you remove the beads and antibodies, and then your cells are ready for the second round of magnetic labeling. A second option step can be applied using the REAlease reagent to also remove the bound antibody biotin complex. So to obtain completely label-free cells and make all the epitopes accessible again. This offers a researcher an entirely new level of flexibility and new suppression strategy. For example, regarding to Tregs. First, pre-selection of CD for positive cells, even from difficult sample or low frequencies by positive selection. Then after removal of the label, as you can see here, cells are ready for the second round of deeper subset separation. For example, we apply here CD25 marker. And this is only one example of a multi-step isolation strategy that combines the advantages of positive selection with label-free cells. I highly recommend you to visit our web page regarding to REAlease MicroBeads and take advantage of this very nice technology. And as the last Treg isolation strategy, I would like to talk about sorting with MACSQuant TytoCell sorter. It's an enclosed and has disposable cartridge. You just need to transfer your cells to the cartridge, run the specific program, and leave the sorter. It gives no stress and pressure to your cells, and according to the microchip-based sorting process, it's completely gentle to your cells. And as I've mentioned before, Tregs are really sensitive and fragile cells. So this device, this sorting machine, is ideal for Treg sorting in both research and clinical scales. And we have very good application note, panels of antibody, and good support in this part for you. After Here I want to show you one example of Treg sorting using MACSQuant TytoCell sorter. You can see here we made a list of antibodies to isolate memory Tregs. And we transfer our labeled cells or stain cells to the cartridge and put it in the TytoCell sorter and run the specific machine, and then we leave the machine for a while. And afterwards, you can see the high purity of your favorite cell subtype. In addition, the viability and quality of the cells after gentle sorting is amazing. You can use your sorted cells for all type of downstream application. And please keep in your mind, whenever you need to sort any Treg subpopulation and using MACSQuant TytoCell sorter, feel free to contact us. We will help you to design the best antibody panel for sorting your favorite Tregs. In the next step, we go to culturing and expansion of your Tregs. For efficient culturing and expansion of your isolated Tregs, we need to consider about three main factors. The first one is a proper cell medium that we offer you TexMax, which is our dedicated T-cell medium. Cytokines play an important role. As you know, the correct dosage, stability, and activity of cytokines are super important to have a good expansion of your target cells, especially Tregs. And for Tregs, we apply IL-2 from MACScytokines that I'm going to explain more. And Treg activation reagents, we offer you Treg Expansion Kits that are very efficient and available for mouse and human. Okay, TexMax optimized T-cell medium is serum-free, free of animal-derived components. It has reliable activation, and it has optimal growth. It actually guarantees optimal growth of your cells with high expansion rates, and we have it up to max GMP quality, and we use this medium for activation and expansion of T cells and Tregs. And in this graph, you can see the comparison between the TexMax medium efficiency comparing to competitors and RPMI, for example, and you can easily see the difference and higher rate of expansion out of TexMax medium application. MACScytokines standardize for highest reproducibility. They have high quality and available in research, premium, and GMP grade, reliable with high lot specific biological activity, flexible. We have them in a small bulk and even custom filling available. They are reproducible, and regarding to time effectiveness, you don't need to have any lot-to-lot testing, and it is really cost efficient because you get lot-specific activity for exact dosage. If you have a look at these graphs, you can see the efficient cytokine usage with specific unit dosing. Green arrows indicate concentration of cytokine input to reach maximum cellular responses. Identical activity can be reached with cytokine concentration at 100 and 1000 IU per ml. Yellow and red arrows indicate insufficient cytokine input. Due to variable cytokine activity, experiments are often oversaturated, as you know. With the given lot-specific biological activity, you can use cytokines at precise concentrations and save reagents. Knowing the exact cytokine activity, you can use the optimal amount of cytokines in your experiment and save up to like 10-folds. Highest quality manufacturing and reliable testing results in a superior biological activity. As you can see, we test human IL-2, which is the mandatory component for Treg expansion. And we have rapamycin in GMP form, which is one of the most important factors in culturing of Tregs. It allows the in vitro expansion of murine and human FOXP3 positive Treg cells, which maintain their regulatory phenotype and suppressive capacity. Here, I would like to show you the efficiency of this combination of the products with some data. After the isolation step using different isolation kits, you can see here that we use different isolation kit. Cells were cultured in Tex-Mex medium plus IL-2, rapamycin, and Treg Expansion Kit. The Treg Expansion Kit are optimized simulation, maintain Treg phenotype and FOXP3, and they are available for human and mouse. And you can easily see the high in after 21 day expansion. You can easily see the high rate of expansion in addition to maintenance of Treg characterization by conserved expression of FOXP3. Okay, more specifically, we had a deeper look on the expansion of MACSxpress Treg isolated yield. You can see here that after 2 weeks, we had 22-fold expansion, and at the end of 3rd weeks, up to 40-fold expansion. For Tregs, maintain Treg phenotype and FOXP3 expression is very important. And in the FACS plots here, you can see that on day 14 and 21, the characterization of your Tregs are completely conserved. You can see a high rate of cells with expression of FOXP3. The last but not the least step in Treg workflow is Treg analysis. The analysis of Tregs can be done in each step of your workflow to check the efficiency of, for example, tissue dissociation, separation, expansion, and so on. And you can analyze your Tregs in 2 ways, phenotypic analysis and functional analysis. For phenotypic analysis, we offer you Tregs detection kit or our recombinant antibodies reaffinities. And for the functional analysis, we offer you a very exact and effective Treg Suppression Inspector kit that I'm going to talk about each of them in the following slides. The various MACSQuant analyzer are fully automated flow cytometry machines, and each of them has different features. For example, all of them has 3 lasers, and you can detect up to 16 optical parameters. And the other example is like MACSQuant X, which is high throughput variant with faster sampling and process for up to 384 well plates, which is really ideal for the teams that are working with several samples and they want to analyze them at the same time and have a good comparison. Okay, you can use our flow cytometry in combination with Treg detection kits, which are optimized antibody kits with pre-titrated antibodies developed as a simple 1 reagent solution and available for human and mouse samples. And let's go through reaffinity antibodies. You know that the most efficient way to reduce background in flow cytometry is to choose the right antibodies. So, reaffinity antibodies are a type of recombinant antibodies that allows you to have a control over the sequences. The FCR binding site was mutated to reduce on a specific background, and the main benefits are like highest purity and lot-to-lot consistency for greater reproducibility, no tedious and costly FC receptor blocking a step for lower background, and they have one universal isotype which control for convenience and cost saving, and they were tested for hundreds of specificities. And you can combine these antibodies and use reaffinity and even our classical hybridoma drive antibodies with proper fluorophores, which names iodized. They are bright, distinct, and hassle-free. And I would like to again invite you to visit our webpage to get more information about each of these antibodies, detection kits, and so on. And the good news is that we offer you different panels of antibody. And if you want to design your own panel, we can help you. We have wide variety of antibodies with different labelings, and we will help you to design your favorite one. In this slide, you can see that we detected our Tregs with different markers such as CD4, CD25, CD128, CD45 RA, CD45 RO, and FOXP3 and Helios as intracellular markers. And you see that we have very few or no background at all, and it shows the high quality of our antibodies. And here I just mentioned to a scientific poster. Please look at the scientific poster, look at the Treg application page on our web page, and get more information and have a better overview about our Treg antibody panels that we cover all subtypes of Tregs for you. Okay, here we can see an application example in which samples collected from the initial lymph node spleen, and we use Treg Isolation Kit CD4 CD25 positive, and different cell fractions was analyzed. You can easily see the efficiency of MELS CD4 CD25 positive Treg Isolation Kit with detection of different cell fraction you see here. And in the next step, all the cells were expanded in the presence of IL-2, rapamycin, and Treg activation kit, and so on. And proliferation analysis of Tregs shows that upon activation of Tregs in the presence of IL-2 and activation reagents and so on, cells have undergone two division by 48 hours. By 96 hours, most of the cells have divided between 4 to 6 times and retained high level of expression of CD25 and FOXP3 markers of Tregs. You can see here. Altogether, these results show how the CD4 positive, CD25 positive Treg isolation kit works, and then this yield can be expanded in a best way in a medium which contains IL-2, rapamycin, and Treg activation kits. For the functional analysis of your Tregs, I would recommend you our Treg Suppression Inspector kit. This kit is the proof of the efficiency of your suppression and expansion step because it shows you the functionality of your final Treg yield. But let's have a look at the concept of this kit. In this kit, we have Treg Suppression Inspector, which is a polyclonal stimulus, and MACSQuant coupled to alpha CD3, CD28, CD2. And these MACSQuants stimulate your T responder cells in an optimized way to proliferate. Let's see how we test the functionality of Tregs with this inspector kit. Here you can see when your responder T cells stimulate, they start to proliferate. But when your responder T cells co-culture with Tregs, after receiving the stimulation, Tregs suppress the proliferation of the T responder cells. And it shows that Tregs has the suppression functionality. In this graph, you can see high proliferation of T responder cells in the absence of Tregs, whereas co-culturing of them with Tregs inhibits the proliferation of T cells. And at the end, you can see that our polyclonal stimulus is optimized for T responder cells and not for Tregs. And at the end, I would like to show you another application example and finish my presentation. Tregs were isolated here from human blood using human CD4 CD25 Treg Isolation Kit. After separation, cells were analyzed by flow cytometry. Then the positive fraction as Tregs and negative fraction as T responder cells were used for the suppression assay. Just mentioned here that you can use the Tregs and T responder cells from the same donor by using these kits. You can see in the results, in the absence of Tregs and presence of a stimulus, T responder cells proliferate, whereas presence of Tregs stop the proliferation of the T responder cells, and you can see the same results when there is no stimulus. Okay, just to remind you, one more time that we think translational and have a complete sort of product in research and clinical scale to support your translational studies. I put these useful pages and source of data for your consideration. You can just click on this link and find your information, favorite information. And in summary, I would like to say one more time that we covered the whole Treg workflow. We offer solution for every pain point. Feel free to contact us. We are more than just cell separation company, and we are translational company. Please visit our webpage and regarding to Tregs, our application page. And if you need more information, all the resources are available online. Just go ahead, have a look, and if you have any problem, any further question, feel free to contact us.